Thank you all very much for joining us this afternoon. This is our 10th one in this particular series of Research Spotlights, and it's great to have Kelvin join us today, all the way from Pennsylvania in the States. Just before we start, I just wanted to sort of tell people that this has been possible today by getting sponsorship, because obviously we can't do any of this without sponsorship, and we've had support from Sumanas Pharmaceuticals in Kyoto, Kirin. So we're very grateful to them for helping support this series as we explore different aspects of Parkinson's disease. And I can see some familiar names already appearing on the list of people attending. For those who haven't been to one of these before, the idea is that we will explore a little bit about Kelvin. We always like to find out where people have come from, how they ended up in their field of Parkinson's disease. And then we're going to get to the sort of main topic today, which is around modeling Parkinson's disease in animals. Is it worthwhile? Is there anything useful we can learn from that? What can we learn from that? And some of the challenges and some of the advantages of that system. And there is no better person to talk about that than Kelvin, who is famous for many reasons, but I suppose is probably best known for his pioneering work using a particular form of alpha-synuclein to induce pathology in animal models called preformed fibrils. But more of that later. So welcome. And if you have any questions, by all means, stick them in the chat and I will try and get to them as well as carry on with this conversation with Kelvin. So do put your questions in there. And just to reiterate, as Eli said, we will be taking pictures. So if you do have your camera on, we will take it that is consent for that picture to be put out on social media, which will only be through the WPC. So Kelvin, thank you very much for joining us this afternoon, or this morning, or this evening, wherever you are. I was wondering if you could just tell us a little bit about yourself. I mean, I'm always interested to know where you began life. So where did you do your undergraduate degree? Then you obviously went off and did, postgraduate PhD. So tell us a little bit about, where you began and then how your career has sort of progressed from there.
Sure, Roger. So it's great to be here. So thanks for having me, first of all. And a little bit about myself. So I actually have a case of sort of mixed or lost identity, I guess. I was actually born in Hong Kong, so former British colony. A lot about it on the news right now. But I actually moved as an infant to Canada and grew up in Toronto. Did my primary schooling there, went back to Hong Kong as a teenager, and ended up for college back in Canada in Montreal. And so I did my undergraduate training there, and I actually initially trained to be a microbiologist. The first week I arrived on campus, the entire dorm went out to see the movie Outbreak. This was with Dustin Hoffman and Rene Russo, and I, committed myself to being a virus hunter. I think I was about 25 years too early for that, and actually ended up staying in Montreal at McGill for my postgraduate studies. And I initially wanted to be a virologist, and just through some unpredicted series of events, the lab that I was supposed to work with actually lost its funding, and I ended up sort of a bit lost and found neuroscience instead. And I'm so glad that happened, because it was the most interesting thing that I could have been studying, and I was just totally immersed in it. And what brought me to Parkinson's disease, I think it was two things. First of all, the person I was working with at the time, my supervisor, was a functional neurosurgeon. And
so this was in Montreal.
This was in Montreal, yeah. So Abbas Sadicott, who was my mentor at the time, did a lot of surgeries, especially, deep brain stimulation was just beginning to really take hold. And so I was fascinated about what was going on. At the time, I was studying how the brain was developing, but it became very clear that, a lot of things were still unknown about how the brain sort of falls apart later on in life. And so that just got me hooked. And I did my postdoctoral studies down here at the University of Pennsylvania, and just haven't managed to escape ever since. But while I was here, I began studying this protein that you were talking about, alpha-synuclein, which is found in Lewy bodies. And one of the things we realized was that it was able to self-propagate, right? And spread inside the brain. And so it sort of came full circle for me. I set out to study sort of these infectious agents or pathogens, and thought I had left that forever. And finally, I'm dealing with something that resembles a pathogen in the brain again. And
so it wasn't that you came to study UPenn specifically around this sort of idea that the alpha-synuclein protein could behave in a sort of infective viral fashion. It was just coincidence that you started working on that, and then it sort of reflected back on your work with sort of your aspirations to be a virologist and how viruses spread from cell to cell.
Exactly. Yeah, it was it was something that was in the back of our minds. A number of people had suggested that as a possibility throughout the past couple of decades, and I think we were just either brave or silly enough to give it a shot and test that hypothesis using an animal model.
Yeah. So let's. Obviously. Animal models, something we all know about with diseases. We're always trying to model what we see in the clinic in the lab, and obviously you've devoted, most of your professional life to trying to develop this and use it to understand Parkinson's disease. So I suppose the question that I'd like to start with is, given that the only animal that gets Parkinson's disease is humans, wouldn't we be better off just studying, people with Parkinson's, rather than trying to model it in the lab? Because whatever we model, it's not going to be what we see in the clinic. So why bother making animal models? I suppose that would be the question. So do you think that, you obviously do because you're doing it, but do you think there's merit in animal models? And if so, what do you think those merits are?
Mm-hmm. Yeah, that's a really good point, Roger. And, I want to first be very clear that none of the models that we have presently and likely in the future are going to be perfect, right? But they do provide us a lot of opportunities to examine aspects of disease and test hypotheses that we otherwise wouldn't be able to do in humans, either for ethical reasons or for physical reasons. So I'll give you an example, which is one of the things that we've been very interested in is what happens very, very early on in Parkinson's disease, right? What are the very, first events that take place? And you can test different hypotheses using cells, using animal models, if you have some ideas you want to test. But that's almost impossible at the moment with humans, with patients, right? By the time we get them diagnosed, it's probably likely that the disease process has been taking place for some time, probably a number of years, if not even decades. So we don't have that window, and the animals, I think, provide that really nice opportunity.
Okay. So I would counter that and say, well, obviously there are genetic forms of Parkinson's disease where you can test people for the gene, and therefore you could say, well, actually, I know that someone's got this gene for Parkinson's disease, so I can start studying them now, even if they're not complaining of anything. So one could, to some extent, use patients with genetic forms of Parkinson's disease. Do you think that's a valid approach, or do you think using genetic forms of Parkinson's disease is, it's only the minority that have clear single genes causing their disease, so therefore it's not really addressing Parkinson's disease itself?
I think, the other thing to keep in mind is they're not mutually exclusive, right? It's not like we have to only study humans or only study animals. And I think they've been really, really complementary. And we've seen over the years genes that have been discovered, single genes that cause Parkinson's, for example. We've been able to identify these genes in humans, do the genetic studies in humans, but then, when we're able to generate animal models based on these genes, right, and show that they do, recapitulate some aspect of the disease, that's been, that validation is really important for the scientists. It gives us, additional confidence that, these genes are really doing something that contribute to disease. And I think, it's that's very valuable. That's very important for the process. Yeah.
Because there's a question that then Stecker sent in, which is, how many genetic forms are there of POG disease? I mean, there are lots of genes associated with POG disease, but I guess, the question is like LRRK2, for example. I mean, if you get LRRK2, you can get POG disease, you can get POG disease with dementia, or you can get nothing at all. So a lot of these genes, I guess, are more risk genes than actual purely causative. But I take your point. If there's a gene associated with a form of POG disease in the clinic and you can demonstrate something in a model, then that gives extra weight that gene is in some way influencing what you see in that condition. So I mean, obviously, it'd be useful to sort of just explore. I mean, there are specific questions here which we'll come on to in a minute. The different types of models, because obviously we're talking about animal models here, and animals. I mean, a lot of the people here are scientists, they'll know about it, but I mean, animals we typically think of when you think of animal models, you think of mice, you think of rats. But some people use, fruit flies, Drosophila. Some people use nematodes, C. elegans. So there's a whole range of animal models, and I suppose it'd be useful from someone who's obviously thought about this and worked with it. Is there a particular model which you think is best suited for studying Parkinson's disease, or do you think all of these different animal models have some merit? And if so, what is it we're exploring with them? Because obviously, if you get a fruit fly that doesn't look so well, I mean, that's very different to what I see in the clinic. So I mean, the argument would be, well, that's great. If I happen to see fruit flies in my clinic, that's terrific, but I don't. So it's not very useful. So I'd be interested to hear how you think these different animal models speak to different aspects of the disease.
Yeah, I definitely agree with the latter, where, I think, most of these organisms that people study have their individual merits, right? And there are some things that, for the typical person out there might not realize. There's a lot of logistics to the science that we do. So, for example, we can study organisms that are closer to humans, right? Mammals, especially higher mammals, or even non-human primates. Those are very close to humans. But in getting that fidelity with human biology, what we lose is, for example, throughput. So we can do thousands and millions of experiments with worms and fruit flies. If you're working with these larger animals, you might be only able to do a handful or a dozen at a given time. So that's one consideration. The other thing.
So the more primitive the organism, the easier it is to screen, which you just couldn't do with screening for drugs that may have an effect. So they're useful in that sense.
Exactly. Yeah, that's definitely one consideration. But it also depends a lot on the exact question we're trying to ask, right? So if we want to look at some very fundamental, process that occurs in all organisms, we could perhaps use a simple organism like a worm or a fly. But if we want to look at very, complex behaviors, for example, it would be useful to look at, more complex organisms as well.
Okay. So if I was to ask you what would be your preferred model? So I mean, and then we're going to come on and discuss the sort of models that you've developed yourself. I mean, what do you think should be the characteristics that best exemplifies the model? Because, as you say, one of the problems with Parkinson's disease is it only happens in humans. It takes decades before it becomes fully manifest. So it's a very slow process. It affects more senior people, all of which creates challenges in the lab. So what would you say is the best models that we have for Parkinson's disease, and why do you think that?
That's a tough one. I think I'm gonna, skirt outside the box here a little bit and say that, I think the best model is actually, a smorgasbord of models, and to have, I prefer having a wide array of tools in the toolbox rather than, one specific tool that we can go to. And this goes back to, one of the points that you mentioned very early on, which is that we don't have a perfect model, and it's unlikely that we ever will, right? And so it would be a mistake to rely too much on a single model. I have my own personal favorites for different reasons, but even those are fraught with pitfalls and disadvantages. Yeah.
So your approach would be to say no one model is better than another. They're all addressing slightly different aspects of a disease process rather than a disease state, if you like. and together they'll give you some information. So I mean, you're probably best known, Kelvin, and I may be wrong in this, but for developing this preformed fibril model of Pugsy. So it would be helpful just to sort of, I mean, most people here I'm sure are familiar with it, but perhaps just explain how that model works and why. Well, you might not think it's a good model, but why you thought it was a good model or still think it's a good model.
Sure. So so the preformed fibril model, it goes back to this concept that proteins, right, can be folded in many different ways. And what it turns out is that in Lewy bodies, which we see in human Parkinson's patients, it contains a protein called alpha-synuclein, and it's folded in a very abnormal way, a very unique kind of fold. And what's special about this fold is that if it comes into contact with other, alpha-synuclein proteins, it will actually convert them to this abnormal form. So it has the ability, sort of like having a bad classmate or something, a bad influence around you, right? It'll convert you to something that you don't or shouldn't be. And what we realized was if we took that form of the protein and injected it into the brains of mice, then it would start this conversion process. And lo and behold, we would start forming things that look like Lewy bodies in the brains of these mice. And not only did they start forming, but they would start transmitting as well. And what was very surprising at the time was that this actually, is consistent with what we see in human disease, which is that through many years of studying Parkinson's patients that have come to autopsy, you can actually see that in their brains, these patterns of Lewy bodies distributing in a way that would suggest that there's some sort of spreading or transmission that's occurring. So that's what made the model very exciting. Initially, it's actually continued to be something that's very widely used. I think the key advantage to the model is that it turns out that it works not only in mice, but it works quite well in just about any organism that expresses alpha-synuclein. So we've been able to work with collaborators to get this model up and running in non-human primates, in other rodents, other people have done marmosets, and so on and so forth. So it's something that's conserved across all these different species. It gives us a lot of flexibility. The other advantage is that because we are injecting this material or introducing it artificially into these animals, it allows us to start this Lewy body formation process in just about any part of the brain, or in fact, any part of the nervous system, both inside the brain and in the periphery. And that gives us a lot of flexibility in terms of trying to test hypotheses about where Lewy bodies begin, where they first form. And one of the things that the field has been doing is seeing if that matches any of the patterns that we see in human patients. And so that's been really exciting. And in fact, a lot of the sort of next generation of these models is that people have been actually taking synuclein, not from a test tube to inoculate these mice or other animals, but actually taking Lewy bodies directly from patients and seeing if that can spread as well. So those are the advantages. There's a couple of disadvantages is that, obviously in humans, most Parkinson's patients don't experience anything like, being injected into the brain with, abnormal proteins. So we still have to find, what is it that drives the initial process in people? And I think that's a big and very important question that we still have to answer in the field.
What about the concentration? So one of the comments here is that, one of the issues is that people inject, and I don't know how this applies to preformed fibrils. Obviously, in the Parkinson's brain, the levels of alpha-synuclein are not abnormal. It's just, it's aggregated into an abnormal form. And so the question is, are you simply overloading the system by adding more alpha-synuclein in whatever form? And so the poor cell doesn't have a chance because it's overexpressing a protein which it's not used to experiencing, and therefore that's what's clogging it up. and therefore its relevance to when it appears to be normal levels in patients is not the case. So it's an artificial system, and one of the questions that's come in is that many grants go down because they don't use this model, but the criticism will be, well, the model is overexpressing alpha-synuclein, which we know is in a case of Parkinson's disease. So I mean, what are your views on that? The levels that you're, and it's not just with the preformed fibrils, but often a lot of the, when people put alpha-synuclein in with viruses, overexpressing in particular transgenic animals. I mean, how relevant are those, and how relevant is the preformed fibril to that argument, really?
Mm-hmm. Yeah, that's a very insightful question, actually. And so let me sort of clarify one thing first, is that one of the other strengths of these injection models that we initially developed was the fact that they don't use genetically modified animals, right? So we're using animals that are non-transgenic, non-modified, and so they express levels of alpha-synuclein that are, what you would consider healthy. And so, by putting a very small amount of these abnormal alpha-synuclein proteins in the system, we can actually instigate this process. It's sort of catalytic, if you will, right?
And I'm right in thinking that the pathology you see in the cell is not the preformed fibrils just all aggregating in the cell. The preformed fibril, as you were saying, templates the synuclein that's in that cell. So ultimately what you see in the cell is an aggregation of the synuclein from the host, not what you've injected. Is that right?
Exactly. So we're putting in, micrograms, and in some cases nanograms or picograms of protein. So it's a very tiny amount, like in relation to the synuclein that's already in the animal. And again, like I said, it works even at physiological levels of alpha-synuclein.
Okay. And that, you would feel, is, for some of the reasons you said, is a better model than other models. I mean, one of the recurring questions that comes up is, obviously the model you have is, as you said yourself, is there to answer a question. And so the model might not be a model of Parkinson's disease, but it might be a model about the behavior of alpha-synuclein, or how does some component in the cell, the proteasome or something, interfere with alpha-synuclein. So I mean, how does your model compare with toxin models, for example, which obviously been around? Well, 6-hydroxydopamine has been around since the 1970s, and MPTP since the 1980s. These are obviously toxins driving cell death. I mean, they would seem to me to be addressing a different aspect of Parkinson's disease, but it'd be interesting to know whether you think they have any merit against the model which you've just described.
Yeah, I think there's a lot of merits with some of these more traditional models. Like you mentioned, the toxin models have been used for decades, right? And they've given us already a lot of very important insights in terms of what are the consequences of destroying the dopaminergic system, for example, and what that process might look like, even though it's accelerated, right? And you have to remember that a lot of these toxins were chosen because they've been observed in real life in humans to have an effect and cause something that really looks like classical Parkinson's as well. And so there's definitely a lot of merit in that. I think if there was a disadvantage to some of these models, or a downside, is that first of all, most of them are very focused on the dopaminergic system, right? And so they don't give us quite as much opportunity to address some of the non-motor aspects of Parkinson's that are increasingly clear that occur in many, many patients. The other thing also is that in most cases, these models don't recapitulate some of the pathologies that we see. So, for example, the Lewy bodies and some of the more subtle pathologies. And so, the question is, again, how close is it to what we see in humans? But I would say it's given us a lot of information as well.
So I suppose, and for those who were on the last one of these research spotlights when we were talking to Marlin Palm and Agneta Kirkby, I mean, the toxin models are quite useful if you're studying dopamine cell replacement, because you're knocking out the dopamine system. So coming back to one of the questions, what's my model designed to do? in the case where you're using toxins, the model is designed to kill the dopamine cells. Can I replace them with a transplant? The answer is yes or no, depending where who you are and what you believe. But that's what the model. But it's not really looking at any aspect of Parkinson's, whereas yours is very much around studying the behaviour of alpha-synuclein, I suppose. So you would say alpha-synuclein is central to Parkinson's, and it's not just alpha-synuclein, because obviously you said that Lewy body extracts can also do the same, which does contain alpha-synuclein, but other things as well. So these models which you use are primarily, I suppose, studying the behavior of alpha-synuclein on the assumption that alpha-synuclein lies at the heart of Parkinson's disease. And that would be the key aspect of your model, rather than I'm really capitulating all of Parkinson's disease. Would that be fair, or do you think that's an unfair comment to say that you're not really looking at Parkinson's disease with your model, but more at alpha-synuclein?
I think, another way to think of it is to think of Parkinson's disease as a alpha-synuclein disease, right? Okay. When you sort of look at all the Parkinson's patients out there, the overwhelming majority, I think it's 98% or 99% plus of them, have Lewy bodies, right? And have alpha-synuclein pathology. So clearly, alpha-synuclein is definitely misbehaving in the vast majority of patients, and so it's not too far-fetched to think that there's something central about alpha-synuclein in that disease. I think the question now sort of goes back to, are we focusing on the correct process that alpha-synuclein is contributing to, right? Yeah. And this is something that I think over time will tell. But what I want to maybe share is both work that we've done, but work that a lot of other colleagues have done over the past several years, is to show that we can actually recapitulate a lot of the both the motor and the non-motor behavior that we see in patients with some of these models. And I think that's giving us, again, not definitive proof, but giving us more confidence that perhaps even in patients as well, the synuclein that's forming these Lewy bodies is driving some of these symptoms that we see. Yeah.
Okay. So if we sort of just drill down a bit on that, so what do you think animal models have told us about Parkinson's that we didn't know, that we would never have known and didn't know with it, and how has that translated? Has any of that translated into a benefit in the clinic to date, would you say?
So, I mean, if you think about the example I keep coming back to is, the discovery of L-DOPA, right? And, it was you can think of it as an inadvertent model of Parkinson's disease. They didn't, the researchers didn't set out to design a model of Parkinson's disease, but that's what they developed, right? And that's given us a treatment that's, withstood the test of time for over half a century now. And I think there are some other things, a lot of drugs that are coming through the pipeline that look very, very promising, very encouraging. But those are going into clinical trials now, and I think we'll see what they bear.
So at the moment, you would say, so obviously that original work I think on dopamine was I think it was reserpine that was used in the 1950s, which depletes dopamine or adrenaline, and the animals became Parkinsonian, and then it led to the discovery of the loss of dopamine. So in that sense, yes, I mean, that's very much laid the groundwork for that. I suppose currently they're sort of unproven, I would suppose, the animal models at the moment, in that they've thrown up various mechanisms which may be relevant, but we haven't yet been able to demonstrate that they are intimately linked to Parkinson's, because we haven't had any data to sort of support that. And I suppose coming back to your model, one of the questions we've had is around obviously antibody therapies. So, the argument will go, if alpha-synuclein spreads from one cell to another, and when it arrives in the new cell, it causes pathology, can I intercept it between cells? And one way to do that would be get the immune system to generate antibodies to kill off alpha-synuclein. I mean, they may even get inside cells. And so far, those trials have not, I mean, there are only a few, but so far they haven't revealed anything that gives us encouragement that that's correct. So I suppose that raises the question: is your model, or are these models? Because I make it sound as though, Kelvin, you're entirely to sort of all models are your models, but but is it that these models are giving us the wrong signal, or are the trials we're doing the wrong trials? And of course, there's no right. It's impossible to know the right question, but I suppose it then brings the question is, coming back to the relevance of the models, and one of the questions is, when should we discard a model because everything we do in the model works, but nothing we do in the clinic makes a difference with that. So I suppose the first question is, do you think these models are misleading us about protein spread, or do you think the trials have given us the wrong, not the wrong answer, but haven't given us an answer yet because we haven't done them properly?
I think it's probably a mixture. I mean, by the way, these are really, really insightful questions. So thank you for asking them. So I think, as I mentioned before, the models are not perfect, right? So there might be aspects of the mechanism of disease that we're missing, and we could be entirely wrong about it, although I feel that it's not likely. And as you also mentioned, it could be that the way the trials are run just didn't give us the best chance of answering that question, right? Of whether, this type of therapy, this approach, in fact, is worthwhile. And so I'll give you one consideration, which is, and this is, we've seen with other diseases as well, whether a therapy is effective or how effective it is depends a lot on which stage of the disease that you administer it, right? So the general rule of thumb is the earlier in disease, the more effective your therapies are likely going to be. Unfortunately, for a lot of these age-related neurodegenerative diseases, whether it's Parkinson's or Alzheimer's and so forth, a lot of the people who are entered in clinical trials have had the disease probably for quite a while. And so, unfortunately, that means that the chances of the drugs working in this population have already been lowered than if they were diagnosed a little bit earlier. So there's still a lot of work that needs to be done in terms of, matching the correct drugs to the stage of disease. Perhaps one of the things that's happening, I think it's quite plausible, is that it's working in these models precisely because we're targeting it. We know exactly when the disease starts. We get to target it when we want. And so we have that luxury that we don't have in the clinic. And so how do we bridge that gap? And I think there's a lot of efforts to try to understand that better, but also maybe to help diagnose patients in earlier stages of disease so that we can bring the drugs to them, at an earlier stage.
Okay, so the idea would be a bit like in the Alzheimer's disease, the earlier you can go, the better. And obviously, if these are fairly innocuous treatments, you could end up just giving everybody the treatment on the ground. It will prevent something happening like sort of COVID vaccination. But I mean,
and I'm being very optimistic too.
I'm now going to throw something at you, which has come up a couple of times in the chat, which is, might you have the wrong end of the stick here? So one of the arguments is that actually your endpoints are the wrong thing that you're looking at. So two questions sort of relating to that. One is that the aggregation of alpha-synuclein is a good thing. And actually inclusions and Lewy bodies are excellent to try and keep the cell alive. So that's one question. So are we looking, actually, when we're looking at aggregation, is that actually a good thing in cells because it's trying to protect them? And the second question is around whether alpha-synuclein is actually just getting rid of alpha-synuclein might be a better thing to do altogether, or that the loss of alpha-synuclein actually it may be or trying to raise levels of alpha-synuclein, the loss of normal function of alpha-synuclein is what I'm trying to say in a rather clumsy way, is actually the problem here. So one is that aggregation of alpha-synuclein is a good thing. So everything you do in your animal models, which is trying to get rid of that, is looking at the wrong endpoint. And the second is that actually the problem with it all aggregating in patients is that you lose the normal function of alpha-synuclein. So what you actually need to do is not get rid of alpha-synuclein, but give them more.
Yeah. So that, again, these are excellent questions. So I think the first question in terms of whether aggregation itself might be positive, right? It might be beneficial. That's that's something we're definitely keeping an open mind about, right?
Yeah.
Again, going back to models and going back to disease, most of the evidence is consistent with when we have aggregation, we see bad things happen. So it's a correlation.
Yeah.
But in the models, at least we can cause aggregation, and we can see that when we have aggregation, it leads to something bad as well. So I would say I would personally lean towards aggregation being more detrimental than it is beneficial.
And can you do experiments where you put something in, like your preformed fibrils, they're going to aggregate, but you stop that aggregation process and the animals get even worse?
Yeah, so we can, and we have done experiments similar to that, and actually quite a number of people in the field have done that. And in most cases, what they see is the animals actually either get better or they stay where they are, i.e. they don't get any worse. So that would, again, point towards the aggregation itself being bad. But again, I think there's still a gap in our knowledge there, is that is aggregation perhaps a preferable fate to something else that would have happened as well, right? And so, that I think the jury's still out on that one.
Okay, so just sort of following up on that, I mean, obviously you developed these models, and as we've said, the models are designed to answer questions. They might not be there to model Parkinson's disease in its entirety, but they give you insights into aspects of alpha-synuclein or whatever. I suppose the question arises that if you're trying to develop new therapeutics, how useful are these models? So obviously, if you've come up with something new in your model, you think, I think it's this pathway. So I think it's the lysosomal pathway or some other way in which alpha-synuclein is degraded. I've got a model, I've got a deficit. I can now screen it to see whether I can make it better. So I can see if you're trying to develop something new, these models could be quite useful. But one of the things we do in the linked clinical trials, which is part funded by the Cure Parkinson's Trust, is we do drug repurposing. And drug repurposing are obviously drugs which are already in clinical use. So the question is, which always comes up every year we discuss this, is how much preclinical data do you need on these drugs before you do a clinical trial? Because if it doesn't work in whichever animal model you're using, should that preclude you from going to the clinic? But we also feel very uneasy just going to the clinic without any preclinical data to support whatever theory it is. So I would be interested to know how your animal models can be used to develop therapeutics.
Yeah, I think it's going to be, we need to consider these case by case sometimes, right? Yeah. And I think if you have a drug, and especially in the repurposing example that you mentioned, I think there's probably a minimal amount of animal testing that's required before going into the clinic. Again, because like you said, we know what these drugs do. We have a lot of data on them. We know their safety profiles. And so that's already mitigated a lot of the risk in giving them to the patients. But I think it's, in these cases, it's nice to have some data, whether it's from animals or from other kind of experimentation, to make sure that, the underlying concept or hypothesis that you're basing this treatment on is in fact valid, right? So for example, if it's a drug that you think is being repurposed because it hits a certain pathway, as long as you're confident that it's going to hit that pathway in humans, and that it's not, overtly toxic or anything, then, whether that data comes from previous clinical data, from previous preclinical data, I think that already helps push things over the line quite a bit.
So for drug repurposing, if I was to say, well, we want to look at a drug, I don't know, like falodipine, which is a drug we look at in Huntington's disease as it happens around autophagy. I mean, in order to use it in a trial with Parkinson's disease, so falodipine is a standard drug used for blood pressure, you would ideally want to see something in an animal model that showed that if it upregulates autophagy, which is a way of getting rid of proteins, at least in the animal models, it does the same and has some effect on the protein aggregation or the alpha-synuclein. That would be the that would be the value of the model, which would just support the hypothesis on how you think the drugs work, which would then allow you to go into the clinic with more confidence.
I think that would be helpful. Obviously, like you said, patients are looking for something. There's no effective treatment. So, the sooner we get things to them, the better. But at the same time, it's not that we have unlimited shots at goal either, right? And so you want to have at least, some level of filtering just to make sure that it's doing what you think it's doing. And at the end of the day, it's empirical, right? It's ultimately it works in the humans or it doesn't.
Okay. So just relating to that is a question which came up earlier on in the chat, which was around, obviously your animal models use animals, so they're not using humans. So one of the questions is, obviously nowadays we can, with reprogramming, you can take human cells, you can reprogram, as you know, into these induced pluripotent stem cells, and now people make these organoids, these small little bits of brain. So do you think there is a merit in combining animal models with a human system, i.e., a human organoid or a human stem cell derived culture, or do you think actually the limitations of these cultures where you just grow things in dishes, or organoids, which are tiny little bits of selective areas of brain, they're not the whole brain, and quite what they contain is a little bit debatable depending what organoid system you've got. So how much value do you put on the need to test what you find in animal models in a human cell line, or conversely, something that works in a human cell, human cells or human organoids, take that into an animal model?
Yeah, I think having these, iPSC derived cells, right? So these stem cell derived neurons from humans, those have been a tremendous tool because, first of all, they're human cells, so that's already taking us halfway to the clinic, right? And even though they are cells, they do allow us to answer a lot of questions. So I think, one of the approaches that we and others have taken is to combine the two, right? To have a combination where we do what we can, we answer the questions that we can using these human cells. And that what it allows us to do is to either reduce or accelerate the work that we end up doing in animal models, right? The other thing it does is, as I used the analogy earlier, which is having more tools in the toolbox, right? If you find a drug, for example, that gives you very encouraging results across many different platforms and many different models, that's obviously going to give you a lot more confidence than if you had a drug that worked only very specifically or on one or two, models. And I think that information is also very important. It helps you predict and perhaps mitigate some of the risks that are downstream, in the drug development process.
Okay, so your approach. So if I was to say to you, look, Kelvin, here's 280 million to find a cure for Parkinson's, and Benjamin Stecker makes the good point that we have very effective treatments for Parkinson's, and I should say that we always talk about disease modification, which is around modifying the disease pathway, but actually in the pre-DAPA era, people didn't live that long, whereas dopamine is disease modifying. It's just not dealing with the disease process. But anyway, that's an aside. So if I was to say here's a lot of money, your approach would be to find a pathway and a therapeutic. You would start off with some human cells, then you would move to your animal models before you'd go to a clinical trial. That's what you would do. And which animal model would you prefer? What would be your preferred cells? What would be your preferred animal model in order to give you this sort of combination that gives you confidence that you're in the right area?
Ooh, that's a tough one. So I think it also depends, rewinding back to the original question, which is, it depends what it is in the disease that you want to address, right? And I'm just sort of playing off what Ben had pointed out, which is that you can address particular symptoms of the disease, right, or particular aspects of the disease, and you could also address something that's disease modifying. I don't think there's I don't think it's worth arguing which is more important. I think both are very important, right? Yeah. And so if you're trying to target a specific symptom or process, then if there are cells that allow us to answer that question, that's where I would go. So one of the I saw Ben's comment.
Not agreeing with you, but carry on.
Yes, and so in that case, again, it's more of matching the question to the tools that we have. So if I wanted, if I was thinking that hitting the lysosomal pathway, for example, is what I want to do, then if the cells allow me to screen for compounds that do that, that would be great. And then we can start layering on, for example, how about cells when they have synuclein pathology? Can we look for compounds that do that in that context, and then move to the animals? I think in this day and age, with the throughput that's possible with cells, that is probably the most efficient approach in most cases. But not all cases would be like that.
Okay, and so I suppose one of the questions that brings up is this sort of reductionist approach. So one of the things I've always enjoyed in meetings is that someone will stand up and say, all the problems in pub disease relate to mitochondrial dysfunction. They give their talk, present their data, sit down. Then the next person stands up and says, all the problems in pub disease are the unfolded protein response. Here's my evidence, so sit down. Next person stands up, it's the lysosomal. And so it goes on. And it could conceivably be true that they are all correct. So I suppose, one of the issues, I suppose, with animal models is the extent to which we should be reductionist, i.e. we pick a pathway and we go with that pathway, or whether we actually say everything goes wrong at some level, and actually our best models are ones where everything is going a bit wrong, of which the primary driver is age, whatever age does. So I would just be interested to hear your thoughts about how we can factor in age into our models, which is clearly the biggest risk factor for getting Parkinson's, and whether we should really be thinking in terms of models targeting individual pathways, whether it be alpha-synuclein spread, a particular intracellular process, or whether we should actually be a bit more looking for everything that goes wrong and trying to correct everything, in much the same way as people found a cure for HIV by targeting, obviously, well, you'd know better than me because you're a virologist at their heart, each part of the virus's pathway. Treatment of hypertension nowadays, you tend to give, ischemic heart disease, you give lots of drugs, TB, lots of drugs. So there's lots of examples where now it's actually multiple treatments which are much better than an individual one. So what are your thoughts on that?
Yeah, I'm glad you touched on that last point, which is this idea of combination in therapy, right? Is that, these are complex diseases, right? There are many things going on. And I think that's why it's been so hard to develop any single model that is in animals, especially, that recapitulates the full disease spectrum in Parkinson's. And there's energetics that are going wrong, there's aging and synuclein and X, Y, and Z. And so this is where we need to consider, if there's so many problems, it's unlikely also that one drug is going to fix everything, and that we'll need a combination like we do for HIV and increasingly so for cancer as well. The question is, how do we achieve that, especially in complex diseases of aging, right? And again, so it's many of these challenges that we've discussed over the last little while. Finding the right therapeutic window, identifying the right pathway, and then doing that in permutations of 3 by 3 different pathways or modalities. So I think there are a lot of smart scientists out there who probably have ways to figure that out from an informatics or from a logic kind of matrix, right? The most efficient way to come to an answer. And I think, what we've been trying to do is just to provide some of the tools. We're just part of the equation, right? I don't think we're ever going to provide that perfect answer, but that's how we see a lot of this animal development. And I think in response to some of the comments too, I want to point out one of the things that we do a lot of is we try to take a lot of feedback from both our colleagues, but also from the clinicians, right? So we regularly chat with our movement disorders specialists here and, at meetings and so forth. And we always ask them, if we wanted, if we're trying to build a better mouse model, for example, what would you like to see? Right? If I gave you a wish list of 3 features that we can recapitulate in a mouse model, what would those be, right?
Yeah.
And we try to also control their expectations, right? And say, like, only 3 things, not 10, not 15, yeah. Right? And it's interesting what they come back with, and I think, as a field, as a community, we need to prioritize some of those things.
Yeah.
There's a lot of resources being pumped into Parkinson's drug development, but we need to still prioritize.
Yeah. I mean, I think your point is extremely well taken, because I think people often think of translation as it starts outside a test tube. It starts, in a test tube, it then moves into a cell, it moves into an animal, and it moves to clinic. And if it doesn't work, that's the end of it. We start at the beginning again, whereas it's clearly a dialogue all the way through. So as you say, things in the clinic. So although the alpha-synuclein trials so far have not provided us with what the answer we want, it may well tell us something which allows us to take it back to the lab to look at it. So, in the last few minutes, Kevin, I just want to pick up on this thing in aging, because your work has transformed how we've understood Parkinson's disease, and that is yet to translate into the clinic, but it's clearly completely transformed how we think about this condition. And one of the exciting things I think that's happened in the last 15 years is, because I'm so old, I've been here forever, that there weren't many new ideas 15 years ago. It was always, can we give a bit more to boost the mitochondria, that type of thing, whereas this has opened up whole new ideas about how proteins spread, seed disease, and one thing and another. And of course it may not prove to be true, but we won't know until we've done all that. But it's really opened up new vistas. But it's this question of aging I want to come back to, because obviously, for those of us who do some research in the lab, you think, well, why don't you just start an experiment in an 18-month-old rat or mouse? They only live 2 or bit years. That's equivalent to an aged person. But of course that costs a fortune to keep the animal going for 18 months before you've done anything. But are we actually shooting ourselves in the foot here? Should actually we stop doing experiments in 3 to 6 month old animals and only concentrate on looking at older animals? Should we actually bite the bullet and only look at aging in the context of disease processes? Or do you think it's perfectly reasonable to use young animals? Because, coming back to an earlier point you made, it may be that the origins of Parkinson's begin much earlier in life than we ever thought. So it's a key. I always find it's a very difficult question to know about aging, because it clearly is so important. But do you think we don't do enough modeling in old animals, or do you think we don't actually need to do that?
Yeah, Roger, I'm gonna again cheat a little bit and say ideally we would do all of the things you just suggested, right? And study both the young animals and the old animals. But you're definitely correct that a lot more attention needs to be placed on aged animals. It's, and they have not been done precisely for the reasons that you mentioned, right? It's expensive, it takes a lot of time. And sometimes, most old mice can still run faster than old people. And so, like
they got twice as many legs.
Right, exactly. So, does aging to whether it's 18 months or 25 months really translate to human aging? And that, is that precise? So those are all things that, don't have a perfect answer at this point. But you're right. I think doing more studies in aged animals, and in fact, a number of groups have shown that even in the models that we use, right, these alpha-synuclein models, that if you now do them on an aged background, that it exacerbates some of the events, some of the pathologies that we see. So it's definitely, aging is a contributor as well.
Okay. So we've only got a few minutes left, but so I want to end up, Kelvin, by asking you what you think goes wrong in Parkinson's. So what do you think is the fundamental problem that causes the condition, and therefore what would be the therapeutic that you think holds the most potential? I'm not going to hold you to this, but I'd just be very interested to know, given all your experiences and working in this field, what you think the primary problem is, and then how and what therapy you're holding out the greatest hope for.
Yeah. So let me qualify my answer first then by saying, I think in my eyes, from where I sit, I think Parkinson's is quite complex in the sense that we should actually think of it like cancers, right? We used to call cancers. but it's actually a whole family of many different diseases. And I think there are these sub distinctions in Parkinson's as well. But I think in most cases, and there are definitely exceptions, but in most cases, I would, put my money that alpha-synuclein is involved somewhere along the way. I don't know, and I don't think anybody really knows how it comes to pass that the protein takes on a abnormal fold. But once it does, we know that from our experience in all of the animal systems that we've had, and generally from what's been observed in the clinic, that once you get that misfolding event, things really take off. Right. And sort of you have this vicious spiral. And so I think from a therapeutic perspective, it would be nice if we can slow that process down. But from a physics and from a chemical perspective, that's very challenging. So we may or may not be able to achieve this, feasibly. But on the other hand, there are a lot of downstream things that occur because of that, such as, like you mentioned, energetics going wrong, so the mitochondria going wrong, protein degradation going wrong, loss of neurotransmitters, so on and so forth. Those are things that we can actually, those are relatively proximal. So those are things that I think are attainable. And so I think that's going to help further improve our treatment. So it'll be a very nice complement, hopefully, for some of the dopamine replacement therapies that are currently there. But the future is always unpredictable, right? Like you said, 15 years ago, we didn't have very fresh ideas. So it's very hard for me to say what's going to happen in 5, 10, 15 years time. But I'm, as I mentioned, I'm optimistic. There's a lot of things that I see coming down the pipeline from other groups, from industry. There's a lot of really great things happening in industry. And I would not bet against all of these things failing. I can't tell you which ones will come through, but I would not bet against all of them failing. So I'm hopeful.
So to sort of summarize it, you think the primary problem is alpha-synuclein, but the most likely therapeutic is going to be downstream of that until we've worked out exactly how we can stop whatever causes alpha-synuclein to misbehave in the first place, which is going to be more challenging. Which I would agree with you. I mean, I think it's easier to go for the downstream than trying to work out how you can affect that process that we don't know. So we're pretty much out of time, Kevin. I just want to thank you for a very, very stimulating discussion. It's been absolutely terrific. You've been fantastic with the work that you've done. It's been fantastic discussing it with you. I would also particularly like to thank our audience this afternoon, who I think have posted more questions than almost any other one of these chats we've done. I'm sorry if I didn't ask your question. It is quite difficult to keep the conversation going whilst also looking at what people have put in the chat. But thank you very much. They've been fantastic comments. It's been a fantastic debate. So thank you all very much for joining us this afternoon. I would just like again to thank the sponsors for this meeting, which is Supernus Pharmaceuticals in Kyoto Kirin, and invite you to all come back on Tuesday, September the 13th. I hope it's not going to be unlucky for Simon Stott, who's talking that afternoon on drug repurposing for PD. Does it work? Which obviously will pick up on a lot of what Kelvin's been eloquently talking about this afternoon, the value of animal models and how we can use them to best decide which drugs to take forward to the clinic. So thank you all very much for joining this afternoon, but a particular thanks to Kelvin for being such an entertaining and educational person to discuss this topic with this afternoon. Thank you very much.
Thank you, Roger. Thank you, everyone.